Chapter 1 is an introductory chapter. It's an overview of nutrition. In this chapter, you will get an idea of the topics that will be covered in more detail throughout the semester. First of all, let's define nutrition. Nutrition is a science that studies the interactions between living organisms, such as humans, and food. There are different processes involved in nutrition. These include ingestion, digestion, absorption, assimilation, and excretion of food. So ingestion is the taking in or the eating of the food. Digestion is when we take that food and break it apart into the building blocks that make up the different nutrients. Absorption is the taking in of those building blocks by cells. Assimilation is using those building blocks to make new tissues and new cells for the body. And then excretion of food is the getting rid of waste products and the parts of the food that were not digestible. Now we come to the term diet. In this case, diet refers to the types and amounts of food and beverages that are consumed each day. In this regard, diet does not refer to restricting calories or restricting food in order to lose weight. So in this class, we're actually going to use diet in both ways, but primarily diet is going to refer to all the foods and beverages that are eaten on a daily basis. So when we talk about specific dieting plans, in that case, then we will be referring to restriction of nutrients, restriction of calories for weight loss. There are other terms that you need to be familiar with. The first is nutrient. So nutrients are the molecules in food that help all organisms, such as humans, to make energy, grow, develop, and reproduce. We have two types of nutrients, essential and non-essential. Essential nutrients are the ones that cannot be made by the body, so they must be consumed. If they are not consumed in adequate amounts, very specific deficiency signs and symptoms occur. Then we have non-essential nutrients, are those that can be made by the body. We also have energy-producing and non-energy producing nutrients. Energy-producing nutrients are going to produce calories when metabolized by the body. And we're going to get a lot more into what energy really means. But energy-producing nutrients are going to provide building blocks so that we can run all the chemical reactions that are going on in the body. Non-energy producing nutrients do not provide calories but do have other important functions within the body. So let's talk about food choices. Why do you choose what you choose to eat? Well, food choices are highly personal. First of all, we start off with personal preference. You choose something to eat because you like the taste of it, right? Whether it be sweet, whether it be salty, if you like the texture of something, it's just what you like. But there also can be a genetic component to it as well. Eating foods out of habit, ethnic heritage or traditions can also influence our food choices. Are there certain foods that are eaten during celebrations? Social interactions. Do you eat certain foods when you are in a social setting? Let's say when you go to the movies, do you always go get popcorn and a beverage or something sweet from the concession stand? And you can do this even though you've eaten supper 30 minutes before. When you go to the movies, you still have to have that popcorn. So we do have sometimes eat during social interaction. Availability, convenience, and economy. How available are the foods that you like? How convenient are those foods? Do you like cooking at home or do you like going out to eat? Is it easily made? Is it something that takes a long time to make? So all of these affect our food choices. Positive and negative associations. Did you eat something as a child that you just didn't like and it stays with you until you were an adult? Do you have a positive association with foods? Did you have a certain food growing up during family get-togethers and you have a positive association with that? Religious dietary laws. With some people of certain faith, there are specific restrictions on what can and can't be eaten. Our emotions can also affect our food choices. Do we eat out of boredom? Do we eat out of depression? Do we eat when we're anxious, when we're stressed? Or is it the opposite? Can we not eat when we're anxious or stressed? Or do we not eat when we are depressed? So all of this can have an effect on our food choices. Our individual values, your beliefs, your concerns about the environment, if you want a specific type of food, if it's raised a certain way, that can affect your food choices. Body weight and image. And looking at the nutrition and the health benefits of certain foods. So do you want to eat whole foods, minimally processed foods, or ultra-processed foods? Now, let me talk about processing a little bit. Processing is when anything is done to a food. So if you go to an orchard and you pull an orange or an apple off of a tree, you have just processed that food. You have done something to it. In this case, it's very minimal. You just picked the whole fruit off of the tree, but that has been processed. So if now you take that piece of fruit and cut it in half, again, you have processed that food. So processing food means how much has it been handled, how many ways has it been changed or manipulated from the time that it was harvested. So if you wanted to eat an unprocessed apple, and truly unprocessed in the sense, you'd have to get a ladder, go to the tree, and just eat the apple as it hangs on the tree. Then it is truly unprocessed. But that is kind of ridiculous, isn't it? It's a little extreme. So really, when we talk about whole foods or minimally processed food, there actually has been some process done to it. Now, ultra-processed foods have had a lot done to them. And the one that I can think of off the top of my head is going to be like Cheese Whiz, cheese in a can. That is definitely an ultra-processed food. How processed you want your food is also going to influence your food choices. Also, the nutrition or the health benefits you think that are associated with that food, whether they be true or not, will also affect your food choices. Foods provide nutrients. Let's take a look at the different major nutrients that we will be discussing in this course. Nutrients are either going to be macronutrients or micronutrients. We're going to look at macronutrients first. So let's go ahead and define that. Macronutrients are required in relatively large amounts. We're talking about grams per day. These macronutrients are going to be organic compounds because they contain carbon, hydrogen, and oxygen. These are going to be nutrients you already know: carbohydrates, proteins, and lipids being fat. So let me go ahead and talk about organic here just a little bit. When we're talking about foods and we're talking about most nutrients, we're talking about organic compounds. All organic compounds are going to have carbon, hydrogens, and oxygen. That's what makes something organic. Now, when we talk about things like minerals, calcium, phosphorus, those are inorganic, and they are inorganic because they do not contain carbon, hydrogens, or oxygen. So this is different than foods that are raised organically. That is a specific type of agricultural management, and there's certain rules that need to be followed. So don't get confused between organically raised foods and the term organic compound as it refers to nutrients. Water is also a major nutrient. Water is inorganic because it doesn't have carbons, but it is made up of hydrogen and oxygen, and you probably know the chemical formula for water, which is H2O, meaning it has 2 hydrogens and 1 oxygen. Now, water is a non-energy producing nutrient because it does not provide calories. We don't get energy from this. We cannot make reactions go because of water. However, water is necessary because nearly all of the body processes, all of the body's reaction are going to occur in water. So those were the macronutrients. We need them in large amounts. So carbohydrates, proteins, lipids, and water. So now let's define a micronutrient. Micronutrients are required in small amounts. We're talking about milligrams or micrograms per day. So what are the micronutrients? All of the vitamins and all of the minerals. Vitamins are organic compounds. Again, they contain carbon. There are 13 different vitamins, and each has a specific and unique role in the body. Now, they themselves do not provide energy, but they facilitate energy release. They're going to help with the reactions within the body. Almost every bodily action requires assistance from vitamins. But vitamins are vulnerable to destruction. So they can be broken down from heat, like from cooking, light, and chemicals. The other micronutrients are going to be minerals. These are the simplest of the nutrients, and they are inorganic because they do not contain carbon. There are 16 essential minerals that are going to be a part of body structures, primarily the teeth and the bones. We're going to get in detail with the minerals later. Now, minerals are indestructible. I can't break them. I can't break them apart, but they can be leached into the water during the cooking process. So you can actually pull the minerals out of food, and they can be lost during cooking. So, and they also can be lost during food processing. Okay, so later again, we will also take a closer look at the minerals. So the human body is made up of compounds similar to those found in foods, primarily water and some fat, with carbohydrate, proteins, vitamins, minerals, and other minor constituents making up the remainder of the body. So body composition of men and women is going to be a little different. So in blue we have water, in this tan color we have the percent of fat, and in this orange color we have the percent of carbohydrates, proteins, vitamins, and minerals. So you'll notice men and women have about the same percentage of water in the body. But when it comes to fat, you'll notice that the percentages are going to be a little different. Women are going to have a little bit higher percentage of fat when compared to men, and men are going to have a higher percentage of all the other nutrients compared to women, so higher in carbohydrates and proteins primarily, but also vitamins and minerals. So there really is a difference in body composition comparing men to women. So we defined earlier energy producing nutrients. So the 3 macronutrients, carbohydrates, proteins, and lipids, are going to be used to provide raw materials for building new tissues and regulating the body activities. In particular, the proteins are going to regulate digestion and energy metabolism, and we'll get into more detail about that in upcoming chapters. Energy is used to maintain body functions and fuel physical work. So how do we get this energy? Well, when we create a carbohydrate, a protein, or a fat, there are bonds that are going to hold all those individual molecules together. So we have bonds holding the carbons together with the oxygens and with the nitrogens. So when these bonds are broken, energy is released. So that energy can be used immediately by the cell, or it can be stored for later use. If more energy is consumed than is needed, over time, body weight will increase. So this is going to be positive energy balance, where I have enough excess to store for later use. But we also have the other side, negative energy balance. So in negative energy balance, not enough energy is getting consumed, so that the body starts to lose weight because we're having to use that stored energy to run reactions and maintain the body. The energy that's released from carbohydrates, lipids, and protein can be measured in calories. And I'm sure you've heard this word before, but what is a calorie? So a calorie is a tiny unit of energy, but it's so small that even a single apple provides tens of thousands of them. In order to make the math a little easier to handle, the calories are actually calculated as kilocalories. So a kilocalorie is 1,000 calories. So this is what is actually used when somebody is talking about the calories of food. It's based on 1,000. So calories or kilocalories are measures of energy. How much energy a food provides depends on how much carbohydrate, fat, and protein it contains. So when completely broken down, a carbohydrate is going to release 4 kilocalories per gram of food. This is the same amount that you get from a protein. So both proteins and carbohydrates give us 4 kilocalories per gram. Lipids, on the other hand, fats, are going to give us 9 kilocalories per gram of food. So we meet the term energy density. So energy density is the amount of energy, or kilocalories, per gram of food. So we have low energy density foods. These are going to be foods that give us anywhere from 0.6 to 1.5 kilocalories per gram. And these are going to include foods like fruits and vegetables. Then we have medium energy density food, which is 1.5 to 4 kilocalories per gram of food. And this will include foods such as, for example, grilled salmon or lean red meat. And then you have the high energy density foods that are going to provide more than 4 kilocalories per gram. These include peanuts, cheese, or mayonnaise. So let's take a look at these two meals. These are kind of similar to the ones that you find in your textbook. If you look at these meals, you'll notice that they are providing the same amount of kilocalories, 1,575 kilocalories. The meal on the left is showing us a high energy density meal, and the one on the right is showing us a low energy density meal. So what foods do we have over here on the left? We see potato chips, and we see a cheeseburger with French fries. These are all foods that have high energy density, right? So you'll notice the size of this meal compared to the size of this meal. So let's look at some of these low energy density foods. So we have a plate over here of vegetables and dip. We have a salad. We have a sandwich. We have some fruit. We have some yogurt. We have some cereal here. So you'll notice the amount of food that is in this meal differs significantly from the amount of food that we see over here on the left. So looking at these two meals, which one do you think is going to make you feel full faster and probably keep you full for a longer period of time? If you look at it, it's going to be the low energy density meal over here with all these different foods to choose from. But they're still giving you the same amount of kilocalories. So that's something to keep in mind as you're planning meals and making food choices. Let's turn our attention to the science of nutrition. The science of nutrition is the study of the nutrients and other substances in foods and how the body handles them. Understanding the nutritional processes will help us to make wiser nutrition decision. Nutrition depends on several other sciences, including biology, biochemistry, and physiology. We have to take all of these together so that we can understand what is happening with the nutrients, how they're being broken down, and how the body uses them. Nutrition is a relatively young science, but has experienced a tremendous growth as scientists start to apply knowledge gained from sequencing the human genome. As we put together nutrition with genomics and molecular biology, this has actually opened up a whole new study called nutritional genomics. Every day we are learning something new about the nutrients and how our bodies are using those nutrients. So how do we learn all of this new information? Well, advances in nutrition are made using the scientific method. The scientific method uses an unbiased approach to examine the interaction of food, nutrients, and health. So this is different from how most consumers are going to get their information about nutrition. A lot of the times you'll base your nutritional choices due to personal experience. I ate this food and I felt better, or I stopped eating this food and I felt better. So that is an antidote. Okay, that is somebody's personal experience. But it hasn't been, or maybe it has been proved scientifically. So how do we prove it scientifically that it is a real process? By using the scientific method. The scientific method uses a number of steps. We have observation, we look at things. Hypothesis is going to be an educated guess on how something works. We then experiment to test that hypothesis and come up with a theory. Was our educated guess correct, or was it incorrect, or was it partly correct? Here we have the steps to the scientific method as presented in your textbook. So we start off with an observation or a question. I ate this and I saw this happen. So my hypothesis, my educated guess would be this. So let's say vitamin C. I noticed that when I took vitamin C, that my cold didn't last as long. So I have a hypothesis. I have an educated guess that vitamin C prevents colds. Okay, so then I need to test that prediction. I need to test that hypothesis. So researchers design experiments and conduct the research to collect the relevant data. What happened? Then we have the results and interpretation. So we gather the data, we have the results from what we did. So we analyze them statistically and then interpret the data in order to draw specific conclusions. So those conclusions could be either the hypothesis was supported, what I thought happened was going to happen, or the hypothesis was not supported. Nope, I didn't see it. But does the process end here? No, because usually at the end of the experiment, we're left with more questions. So if the hypothesis was not supported, why was it not supported? So this leads to new observations and new questions. So now we take that back and we form maybe a different hypothesis or tweak the one that we originally had. So if the hypothesis was supported, why do we think what happened? So we develop a theory that integrates conclusion with those from numerous other studies. And that's the key to the scientific method. And the key to science is, can that study be replicated? And can somebody else, somewhere else, get the same results that you got? So then we have a theory, but it usually doesn't end there. So we not usually want more detail. Why did it happen? Not only we saw it happen, but why did it happen, or how did it happen? So that also leads to new observations and questions, leading back to a new hypothesis, new predictions, and we have more and more experiments. So you see, the scientific method really doesn't have a start and stop point. It's really almost circular, because once you get the results and find out whether your hypothesis was supported or not, then it always leads to more questions. How do scientists do this? How do scientists and researchers conduct a project or conduct an experiment in such a way that they're confident that their results are valid? Well, first of all, research studies are going to use controls. So if you're doing a research project, let's say again we want to deal with that vitamin C. What I would do is I would have two groups of research subjects. One group of people is going to receive the treatment. They're going to get a vitamin C tablet. Then I have another group of people who are going to be the control group. They are not going to get that treatment of vitamin C. So the control group is really key in these research studies because they're like the baseline. We're not giving them anything, so we need to see how their bodies or how their systems are going to react and compare that to the group that did receive the treatment. So by having that control, that really shows, did the treatment have an effect or not? So how do we put people in either the experimental group or the control group? In the best studies, it's done through randomization. So in randomization, subjects are chosen just randomly, and they're just put in their treatment groups randomly, either by flipping a coin or any other thing that is going to be done by chance. So if I had 100 people and I needed to separate them into two groups to do this vitamin C study, and I didn't have randomization, I could end up with a group of 50 people that really liked citrus fruit. I could have them in the treatment group, and I could have 50 people who really don't like citrus fruit end up being in the control group. That's a problem because we already have bias by having all the people who like citrus fruit in one group. So what I need to do is I need to make sure that these people are randomly put into groups so that I take away that bias or that chance that something is going to work out the way I want it to because of the way the people were put in groups. So randomization is very important in research studies. Sample size is also important. To ensure that chance variation between the two groups does not influence the results, the groups must be large. So let's keep with vitamin C. Let's say that our hypothesis is vitamin C prevents colds. Now, I have 10 people. 5 people taking vitamin C and 5 people in the control group. The 5 people taking vitamin C, let's say one person always gets bad colds. Let's say they get a bad cold every other month. If 1 out of 5 gets a lot of bad colds, that is going to bring down the average of how many people get bad colds. So it could really affect the results by saying vitamin C does not prevent colds. But if I had 1,000 people, 500 people in the vitamin C group and 500 people in the control group, now if one person has bad colds very often, it's not really going to affect the average as much as if there were only 5 people. So sample size is very important in controlling that variability. So when you see commercials that say clinical trials, showed this, have you ever looked up one of those clinical trials? Usually you can find them. I did that for one supplement that was being advertised quite a lot. So I managed to find the clinical trial. The clinical trial had 8 people. That is not a very large sample size. So really sample size, you're talking about hundreds of people or even thousands of people. So then we move on to the topic of placebos. So a placebo doesn't have any medication in it, doesn't have any sort of treatment in it. A placebo is basically a sugar pill. But there is something called the placebo effect, and the placebo effect is a real phenomenon. When people believe that they are taking a treatment or a medication or a supplement and believe that it really is going to help them, it does. But it's not because of what they took, but because of the mindfulness and the physiological processes that are happening in the body due to the brain thinking that it can. So this is known as the placebo effect. So with controls and randomization and a very large sample size, this does help to control the placebo effect so that you do have a realistic answer that if the treatment did work or didn't work. So to control with the placebo effect, we have what are called blind experiments. So in blind experiments, this means that the subjects, the ones who are receiving the treatment, have no idea what group they're in. So the 500 people that are receiving the vitamin C treatment have no idea that they are in the vitamin C group and vice versa. Researchers are then able to collect information with the idea that the subjects had no idea what treatment they were getting. So they're actually measuring real physiological responses. Now, an even more controlled experiment is called a double-blind experiment. So in a double-blind experiment, the subjects do not know what treatment they're on. They don't know what treatment they're getting. And the people that are giving the treatment do not know it either. Because sometimes we give away information from just our facial expressions. You've seen that before. It is possible that when somebody is handing a treatment to a person, let's say they're handing a pill that could either have vitamin C in it or not, that they give away, if they're giving out a vitamin C pill, through a certain facial expression. then the test subject can pick up on that facial expression and goes, oh, I'm getting the vitamin C, so this is going to prevent colds. So by having a double-blind experiment, the person who is giving out the treatments has no idea what they're giving out. So usually the treatments are going to be the same size, the same color, have the same texture, so that there is nothing that gives away whether somebody's on the treatment or not. So all of this is done so that when the data is collected, it is due to true physiological changes and not the placebo effect. Not all research is conducted in a laboratory. We do have different types of research. We have epidemiological studies. So what falls under epidemiological studies? These include cross-sectional studies, case control studies, and cohort studies. So these are going to have strengths and weaknesses. So the strengths of these types of studies, they can narrow down the list of possible causes, and they can raise questions that researchers can pursue. Now, some of the weaknesses include that there's no control over the variables that can influence the development or the prevention of a disease and cannot prove cause and effect. So here we're looking more for correlations. We saw this and we saw this. In this group we saw this, but we didn't see that. So there really is no cause and effect, and we'll talk about cause and effect here in a bit. So what are some of the types of these, some types of this research? Well, in the cross-sectional studies, researchers observe how much and what kinds of food a group of people eat and how healthy those people are, and their findings usually identify factors that might influence the incidence of disease in various populations. So an example of this is the Mediterranean diet. So many people in the Mediterranean regions, they drink more wine, eat more fat from olive oils, and yet have a lower incidence of heart disease when compared to other groups. Here you can say that increased intake of olive oil was seen to decrease the incidence of heart disease. But notice we cannot say that by taking or eating more olive oil, it stops heart disease or prevents heart disease. All we can say that it decreases the incidences of heart disease. So then we have case control studies. So in the case control study, researchers compare people who do and do not have a given condition with people of similar age, gender, and other key variables so that the differences will stand out. Then we have cohort studies. So in cohort studies, researchers analyze the data collected from a selected group of people, or a cohort, over a specific period of time. So they take one group of people and then follow their history over a specific amount of time to see any changes or to track any changes. And this graph over here on cohort studies shows how a certain group of people were followed over time and their blood cholesterol levels were measured over time and how that correlated with heart attacks. So with this graph, you can see that over time blood cholesterol levels increased, and as the blood cholesterol levels increased, we also saw an increase in the number of heart attacks. So again, this is going to be a correlation. They're not saying that increased blood cholesterol causes more heart attacks. You have to be very careful when you're reading these as to the wording. We also have experimental studies. Experimental studies can be broken down into either laboratory-based animal studies, laboratory-based in vitro studies, or human intervention, which are the clinical trials. So again, they're going to have strengths and weaknesses. So the strengths include controlling the conditions for the most part. They can determine the effects of the variable that they're measuring because they can control the conditions. And they can apply some findings on human beings to some groups of human beings. Weaknesses include they cannot apply the results from test tubes or the in vitro studies or animals directly to human beings because there are going to be differences or some differences in the animal physiology when compared to the human physiology. And they also cannot generate findings on human beings to all human beings. So if a clinical trial was being conducted on men from the age of 50 to 70, those results cannot necessarily be applied to women who are 30 to 45. Also, cannot use certain treatments for clinical or ethical reasons. So what the researcher may want to do with an animal cannot be done to a human for ethical reasons. So there are some weaknesses to these types of experiments as well. So laboratory-based animal studies are when researchers can feed animals very special or specific diets that can provide or omit specific nutrients and then observe any changes in their health. So here we definitely see where the nutrient can be controlled. So laboratory-based in vitro studies, what we're doing here is taking individual cells or taking an individual tissue, applying a treatment, and seeing how that tissue is going to react, seeing how those cells are going to react. But again, in these studies, it's very hard to take the results from an in vitro study and apply it right to a human. Why? Because in the in vitro studying, I'm using a very specific tissue or very specific cells. But when we're talking about the human as an entire entity, we have all these different organ systems, all these different tissues, all these different enzymes, all these different hormones that can affect the entire organism. And then finally, the human intervention or clinical trials where researchers ask people to adopt a new behavior. So if we're still staying with our vitamin C, to eat a citrus fruit or take a vitamin C supplement and see what happens. So in clinical trials, the researchers try hard to manage the treatment or the variable that is being measured. So once a researcher collects all their data, then they're going to analyze it statistically and see if anything happened. So we can find correlations. So a correlation only shows an association. Okay, so we can have positive correlations, negative correlations, or no correlation whatsoever. And that's what this graph down here is showing. So when we have a positive correlation, it means as one variable went up, so did the other. So they both went in the same direction. So this is not necessarily a desired outcome. So with our vitamin C, this would say, as vitamin C increased, as the amount of vitamin C in the diet increased, the number of colds that a person got increased. That's a positive correlation. But was that the outcome that we wanted to see? No, because our hypothesis was that vitamin C prevented colds. In this case, in the positive correlation, we found out that vitamin C encouraged colds. That's positive correlation, but that's not what we were looking for. So then we have a negative correlation. So in a negative correlation, As one variable increased, the other decreased. So in this case, as the amount of vitamin C went up, the number of colds went down. So that would be good for our hypothesis, right? So this is a negative correlation. As one goes up, the other goes down. And then we come to this graph over here. So looking at these data points, looking at these X's, can we find any sort of specific pattern to this? No. And because we cannot find a specific pattern, this would indicate there was no correlation. Vitamin C had absolutely nothing to do with colds. Okay, so these are correlations. Again, they're only showing association. They're not showing cause. So this is another thing to be wary of or be aware of when you are looking for information on the internet about nutrition and about certain nutrition studies. It's very hard to say something caused this. Most of the research is going to have correlation. As this went up, this went down, which suggests that there could be a cause and effect. But cause and effect, we're really talking about mechanism. If vitamin C causes fewer colds, what is the mechanism for that? Is vitamin C destroying the viruses that enter the body? Is vitamin C helping our individual cells to fight off those viruses? That's mechanism. And how is the vitamin C doing that? Is the vitamin C getting the cell to make more of a specific enzyme that fights off the virus? That is mechanism. That is what needs to be found for this causes this. So a lot of studies are going to be more of correlation. We saw this increase and we saw this decrease. But until they find mechanism, they really cannot claim cause and effect. Researchers are typically going to be very cautious in their interpretations and their conclusions until there is an accumulation of evidence. Did other labs conduct this same experiment and get the same results? So as more evidence accumulates, then it gives a stronger argument to that correlation and possible cause. Now that a researcher has conducted an experiment, analyzed the results, and has come up with a conclusion, now what? Mostly it's going to be published. So the findings from a research study are going to be submitted to a board of reviewers that are composed of other scientists who are primarily in the same field, who are rigorously going to evaluate the study to ensure that the scientific method was followed. And this is known as peer review. Peer review is going to give validity to a study. So if a researcher submits their work and those who are reviewing it find problems with the research method used or the statistics, how they were analyzed, they are going to deny the publication of that study. And that would actually be sent back to the researcher, who can then possibly fix what went wrong or fix what was weak within the process. And if they fix that, if they repeat the experiment, keeping those corrections in mind, then on the next time around, it could possibly be approved through the peer review process and then published in a journal. So sometimes you'll hear people talking about an article from a peer-reviewed journal. this is what they're talking about. The research has been submitted for review, and it was accepted based on scientific merit. Now, researchers or the reviewers are also going to keep in mind the funding source, recognizing that financial support or if there are other conflicts of interest that may bias the scientific conclusions. Newly published findings, let's say somebody has done an experiment measuring a nutrient in a specific way that has not been done before, those are going to be preliminary and usually don't mean anything alone. So the findings of a study are either going to be confirmed or disproved through replication and reanalysis. Scientific conclusions aren't based on one study, or scientific facts aren't based on one study. That study has to be replicated over and over and over and over again by multiple labs. So if that research has been replicated and replicated and replicated, and each time with that replication, everybody gets the same results, and the analysis shows the same thing over and over and over again, then that gives strength to that conclusion that it is a valid and realistic conclusion. But if through replication and reanalysis it is disproven, then there could have been something wrong with the methodology. Maybe there was something unique that happened in that lab that couldn't be replicated in any other lab. So, one research study on a new topic does not mean really anything alone. It has to be validated through replication and reanalysis. Most of us are not going to become research nutritionists. So what do all these studies mean for us? Using the results of thousands of research studies, nutrition experts have produced a set of standards that define the amounts of energy, nutrients, and other dietary components that best support health. And these recommendations are called the Dietary Reference Intakes. You'll see this abbreviated DRI. So they're the standards defined for, again, energy, nutrients, and other dietary components. And this is actually a collaborative collaborative effort between the United States and Canada. So these recommendations are actually going to be for healthy people. So recommendations can be different for specific groups. So if we're dealing with a group of people who are diagnosed with kidney disease, the recommendations are going to be different for them, because these dietary reference intakes are always going to be for healthy people. Here are 4 other terms you need to be familiar with. Estimated average requirements, abbreviated EAR. Recommended dietary allowances, abbreviated RDA. You may have heard this before as recommended daily allowance. Adequate intakes, abbreviated AI. And tolerable upper intake levels, abbreviated UL. So the estimated average requirements, this is the average amount of a nutrient that appears sufficient for half the population. So the requirement for a nutrient depends on how much is needed on the diet, and the requirements are going to vary due to gender and age. So a toddler is going to have different nutrient requirements when compared to somebody who's 75. And a pregnant woman is going to have different nutrient requirements when compared to a man of 36. The recommended dietary allowances are the average daily amount of a nutrient considered adequate to meet the known nutrient needs of practically all healthy people. So the recommended dietary allowance is actually going to be greater than the EAR, and we're going to see that here in a second. Adequate intakes are the average daily amount of a nutrient that appears sufficient to ensure nutritional adequacy. So there are AI levels designated for certain nutrients, particularly the vitamins and minerals, because there is insufficient scientific evidence to determine an EAR and therefore a RDA. So we know we need these, but we're just not positive in what amount on a daily basis. And typically it's because the amounts are so small that it's hard to do a really good controlled scientific study on them. And then finally, the tolerable upper intake levels. These are the maximum daily amount of a nutrient that appears safe for most healthy people. The intake of nutrients beyond this upper limit or upper level can increase the risk of adverse health effects. Now, typically, the upper intake levels are not going to refer to nutrients from food. What they primarily are dealing with are large doses from dietary supplements or from fortified foods. Let's compare what's happening with EAR and RDA. An EAR has to be calculated and established before the RDA can be recommended. Let's see why. The ERA, or the estimated average requirement for a nutrient, is the amount of that nutrient that meets the need of about half of the population. So let's take a look at this graph right here. So this red line is showing me the ERA of a specific nutrient, and this is just calling it nutrient X. If the RDA was set at this same level, this half of the population would be deficient in that nutrient, because this is showing just meeting the needs of half of the population. So in order to make sure that the majority of the population has adequate intake of that nutrient, the RDA is going to be set well above the EAR so that it's meeting the needs of about 98% of the population. So now that recommended level went from a EAR of about 45 to an RDA of about 63 over here. So now at this level, 98% of the population is going to have adequate intake of that nutrient. So you can see why the estimated average requirement needs to be calculated first in order to get a reasonable recommended dietary allowance for that nutrient. In addition to the recommendations for individual nutrients, there's also a recommendation for energy. And while the recommendations for nutrients is pretty generous, the estimated energy requirements are not going to be as generous. So what is this? The estimated energy requirement, or the EER, is the average energy intake in order to maintain energy balance. So energy balance so that somebody can maintain a healthy body weight, and also to include physical activity. In this case, balance is going to be the key for the energy recommendation. Enough food energy is needed to sustain a healthy and active life. But because any amount of excess energy will lead to weight gain, no upper limit has been determined, because it's going to differ from person to person. So it's hard to say that you can't have, or this age group can only have this much, because if we go any higher, then we're going to start gaining weight. But it's so individualized that really no upper limit for energy has been determined. Although the intent of nutrient recommendations seems really simple, they are the subject of much misunderstandings and controversies. Let's look at the following facts to help put these nutrient recommendations in more perspective. So first of all, these estimates apply to healthy people. They need to be adjusted for medical problems, malnourishment, or other conditions. So somebody who's dealing with kidney disease or somebody who is dealing with cardiovascular disease, their nutrient recommendations are going to be different than somebody who is healthy and doesn't have those conditions. The recommendations are not minimum requirements, nor are they necessarily optimal intakes for all individuals. Recommendations target most of the people, but can't account for individual variation in nutrient needs. And you've probably noticed this. And most of these nutrient goals are intended to be met through a variety of foods whenever possible, because excess intake of vitamins and minerals is very unlikely when they come from food. But using dietary supplements in order to meet the nutrient goals can raise the risk of toxicity. Recommendations also apply to average daily intakes. So trying to meet the recommendations for every nutrient every day is difficult and unnecessary and probably unrealistic. The length of time over which a person's intake can deviate from the average without risk is going to be variable for each nutrient, kind of depending on how the body uses and stores the nutrient. So if you didn't get your full RDA of vitamin C today, there's really nothing to panic about, because maybe tomorrow you'll eat more citrus fruit than you did today, and it kind of all balances out on the average. Also, each of the DRI categories does serve a unique purpose. And the example used in your book is that the EAR are most appropriate used to develop and evaluate nutrition programs for groups of school children and, military personnel, while the UL serves as a reminder to keep nutrient intakes less than the amount that can increase your risk for toxicity. What happens when a person doesn't consume enough or consumes too much of a specific nutrient or energy? If the deficiency or excess is significant over time, the person experiences symptoms of malnutrition. Mal, think bad. So malnutrition, we're talking about bad nutrition. And typically when you think malnutrition, you think undernutrition. But overnutrition is malnutrition as well. You're getting too much. So what are some of the symptoms of undernutrition? So with undernutrition, the person can be very thin, they're losing muscle mass, they can experience things like diarrhea, skin rashes, fatigue, things like bleeding gums, losing hair, problem with nail brittleness. So there are a lot of symptoms that can go on with undernutrition. However, one needs to be careful because these symptoms can also be for other conditions. Overnutrition, we can see conditions such as heart disease and diabetes. What does a trained healthcare professional use in order to find out if somebody is dealing with malnutrition? Well, first of all, they have to get a total picture of the individual using nutrition assessment. Now, there are going to be 4 parts to this nutritional assessment. First of all, they're going to have to gather historical information. What is this person's health status? What is their socioeconomic status? Do they use drugs? Are they on specific medications? Also, they're going to take a diet history. How is that person eating over several days? What are their portion sizes? All of that information would then be put into a computer software, and a nutritional analysis would be done. That healthcare professional would also take anthropometric measurements. What are these? Basically height and weight. They would use these to track and identify trends. Are they gaining weight over time, or are they losing weight over time? A physical examination will also be conducted, looking at the hair, the skin, the eyes, the tongue, and the fingernails to see if there are any specific changes. Is the hair brittle? Are the fingernails brittle? What does the skin look like? What do the eyes look like? And lastly, but definitely not least, are laboratory tests. So blood work and urine analysis to see what is happening within the blood, what's happening internally that might be reflective of a nutrient deficiency. This graph is showing how the individual assessment process will reflect what is happening in the body. So through diet history and health history, the healthcare professional can see if there's a primary deficiency caused by an inadequate diet, or if there's a secondary deficiency caused by problems inside the body. So as we go through this semester, you're going to find out that the liver is an important organ when it comes to nutrition. If somebody is experiencing liver problems, let's say cirrhosis of the liver, that can actually cause a secondary deficiency. And it's not necessarily a deficiency of a nutrient, but there is something going on with an organ that is causing a secondary deficiency. The laboratory tests are going to show declining nutrient stores. So you'd see this over time. You'd also see if there are any abnormal functions happening in the body. Let's say that over time, the levels of a specific enzyme start to change, and that enzyme is necessary for the breakdown of certain nutrients. You cannot see that, but it will show up in blood work. And finally, doing that physical exam, you can see if there are any physical signs or symptoms. Typically, the physical signs and symptoms are going to occur much later than what we see in the blood. So you can actually see what's happening with nutrient levels in the blood first before there are actually any overt symptoms of that nutrient deficiency. In addition to individual nutritional assessments, researchers can also use surveys to do a nutritional assessment of populations. The data that's collected then can be used by various agencies for a lot of different purposes, including the development of national health goals. What kind of surveys do? They can do a national nutritional survey, in which one survey is just going to collect data on the food types and amounts that are being chosen. Another will collect the anthropometric data about people, height and weight. And then this information can be used by government, scientists, or the food industry. So an example of it being used by the government is to find out where high risk groups are, so that maybe there need to be more educational programs in those areas, or food assistance programs, or the regulation of the food supply. Also, we can find out where all the different food deserts are. So food deserts are going to lead to food insecurity. So the food industry would also use this information like in the development of new products. The information from those surveys can also be used to set national health goals. And the program that's going on right now is called the Healthy People Program. So this sets priorities and guides policies that can help increase the quality and years of a healthy life and help to eliminate health disparities. So this is supposed to start at the beginning of each decade, and the program is supposed to set goals for improving the nation's health during the next 10 years. So nutrition is one of the main topic areas, and it's going to have a lot of different objectives. So these surveys can also help identify national trends. How has eating changed over the decades? Well, first of all, we do know that portion size has increased over the decades, and we've also seen an increase in the incidences of overweight and obesity. Foods play a vital role in supporting health. Early nutrition research focused on identifying the nutrients in foods that would prevent common diseases such as rickets and scurvy. However, more recent research is now focusing on food's role in chronic diseases. Chronic diseases are responsible for 7 out of 10 deaths among U.S. adults and account for more than 85% of U.S. healthcare costs. So let's look at this graph. Red is diet-related, yellow is alcohol-related, and blue is non-diet related. So let's look at some of these causes of death among U.S. adults. You can see that heart disease and cancer are going to be the leading cause of deaths. We also have stroke. And if you'll notice, these are going to be diet related. Other diet related include diabetes, kidney disease, and hypertension. Most chronic diseases arise from multiple factors over many years. So a person who died of heart disease may have actually been overweight, have had high blood pressure, been a cigarette smoker, and spent years eating a diet in high saturated fat and getting very little exercise. When we talk about chronic diseases, we also have to keep in mind risk factors. Risk factors either increase or reduce the risk of developing a chronic disease. Now, a risk factor means that there is the likelihood of developing a disease. It doesn't mean if you have this risk factor, you will develop the disease. But if you have this risk factor, you are increasing your likelihood of developing a specific disease. On the other hand, the lack of a risk factor doesn't mean that you're not going to get the disease. It just means that the likelihood of you developing that disease is going to be lower. Risk factors are going to persist over time. If a person in their 20s has high blood pressure, when that person reaches their 40s, they're still going to have high blood pressure. But if that person in their 20s goes to the doctor and has intervention, then their blood pressure can be controlled. So risk factors also tend to cluster. So a person who is overweight or obese may not be physically active. They may also have high blood pressure and have high blood cholesterol. So all of those factors together are going to have that person more likely to develop heart disease. Okay, so you just can't take one and say that it's going to lead to something. Usually they're going to be in a group, they're going to cluster. So what are some health behaviors that people in the U.S. still engage in, knowing that they can be risk factors for chronic diseases? Well, 20% of the population smokes cigarettes, 35% consume more than moderate amounts of alcohol, 50% are physically inactive, or 60% are overweight or or obese. Now, these behaviors can be changed. They can be modified. And as they are modified, they can start decreasing your risk factor for chronic disease. So if somebody is smoking cigarettes, they can try to stop smoking, even though I do know that the cessation of smoking is very, very hard. It's very, very challenging. Many people stop and fail, but trying to stop smoking will decrease a risk factor. Drinking alcohol in more moderation. Trying to become more physically active. It doesn't mean that you have to get up off the couch and just go run a marathon. It just means trying to become more physically active. Walk a little further. Park further away from the door of the grocery store where you have to walk a little further. And trying to lose weight if somebody is overweight or obese. And I know this can also be very challenging. You hear it almost everywhere. All you have to do is lose weight. All you have to do is lose weight. But losing weight can be very challenging. And it can also be very frustrating. But the rewards for losing weight, even if it's just a little bit of weight, can help decrease risk factors for chronic diseases. Now, there are other risk factors, including genetics, gender, and age, and these risk factors cannot be controlled. Okay, so you're going to get older, and you can't control that. Gender, genetics, all of these are basically out of day-to-day control, where you could just change something in your diet and it would affect your genetics, your gender, or your age. That just doesn't happen. So these health behaviors can be modified to lower the risk of developing a chronic disease, while these specific factors cannot be modified to change the likelihood of developing a chronic disease. That's the end of the information for Chapter 1. Remember to read Highlight 1 of Nutrition, Information, and Misinformation.